So I realize my talk title is different than it was in the program, and I'm primarily going to talk about biospecimens. But if you have other questions about in vitro diagnostics, feel free to ask me at the end of the session. As Stephen said, I'm the director of the personalized medicine staff in the Office of In Vitro Diagnostics, where we worry about the clinical laboratory tests that the clinical labs in the United States use. And biospecimens are becoming more and more of an issue for us, and we're having more and more scientific and regulatory interest in them. Just a little background on devices. FDA does regulate medical devices. We were given that authority in 1976 by Congress in the medical device amendments to the Food, Drug and Cosmetic Act. And our requirements have a number of requirements for a test, including these here, which I won't go through. We do pre-market review of almost all tests that are in commercial distribution, either through a 510K moderate risk package or a PMA high risk package. Doesn't matter what you submit, we ask the same type of scientific questions. We look for analytical performance in these tests. That is, how well does the test measurement system actually work? Is it stable, robust, reliable, accurate, and so on? So we look for studies that assess the accuracy of the test compared to a gold standard if possible, often compared to a silver standard, a bronze standard, a pewter standard, sometimes a tin standard. We look for precision of the test. That is, can you get the same result if you run the test over and over and over again? How analytically specific it is. Does it have cross-reactivity or interference issues? How analytically sensitive it is? What's the limit of detection if it's a quantitative test?